speak in front of you because of the things that not only that she has accomplished, but the investment that she has made in herself, in her school, in her community, in her students, in her fellow faculty. I'm telling you, I was so impressed. So I'm going to just read a brief summary that doesn't begin to express how much she has been involved in making the world a better place. So let me introduce Dr. Lindsay Rhodes. Dr. Lindsay Rhodes is a PhD, I'm sure I'm saying that wrong, is an associate professor of biological sciences at Florida Gulf Coast University, where she serves as a leader in cancer research and education. She earned her PhD in biomedical sciences from Tulane University, and she's built a distinguished career in identifying natural essential oils and compounds with anti-cancer properties, particularly in the treatment of breast cancer. Her research has been published extensively in peer-reviewed journals and has garnered national recognition for its impact on cancer therapies. Dr. Rhodes is not only a pioneering researcher, but also a dedicated educator and mentor, guiding students through hands-on research projects that inspire the next generation of scientists. She actively advocates for diversity in STEM fields and is a fellow of the prestigious I Aspire Leadership Academy, which aims to develop senior leaders in science and technology. Beyond her academic and research pursuits, Dr. Rhodes has contributed significantly to community outreach and cancer awareness, making her a truly inspirational figure in both science and education. Please welcome Dr. Lindsay Rhodes.
I'm not sure I can follow that. This holds up. You guys see that okay? You can hear me all right here?
Yeah.
Yes. Great. So I am Dr. Lindsay Rhodes, and I've been at Florida Gulf Coast for about 10 years now. I was telling a few people as we were coming in today that I profess as a profession, so I talk a lot. So you might have to cut me off at 20-25 minutes, and I'm happy to stop talking anytime. But what I wanted to do was just talk to you a little bit about my research, which we'll talk about at the end, but I wanted to give you a little bit more of just kind of a background about cancer in general and then breast cancer a little bit more specifically, and I'm more than happy to take any questions at the end or throughout. So I thought we would just start with a few cancer statistics. I get really excited talking about cancer, which seems weird, right? Because I find it so fascinating. But at the same time, I really know the impact that this has on folks in this room, folks in our lives, and each of us individually. I know quite a few have been probably personally touched by this. So in the United States alone, for this year, 2024, it's estimated to have over 2 million new cases of diagnosed cancers and over 600,000 deaths related to cancer. That's a big number, but this was even more kind of impactful to me. That's 4 cases every minute and 1 cancer-related death every minute. So again, we know that this is a big problem and affects a lot of people. We know we also have investing a lot of time and money into researching cancer and cancer-related therapies. So if we look over time, this goes back to 1975, when we look at cancer incidence, and this is all cancer across the board in the U.S., we see incidence go up and then it kind of flatlines, but we definitely don't see it going down. Why do we see these peaks and these little increases? It could be due to advanced technology, better diagnoses. It could be because we have increased mutagens and carcinogens. And what it really indicates is our lifespan is also getting longer, right? As we have better therapies for cardiovascular diseases and obesity and things that would typically have decreased our life expectancy, we're living longer. And does anybody know what one of the top risk factors for cancer? Age, right? So the longer we live, the more likely we are to gain mutations that will ultimately lead to the disease of cancer. Here's some of the good news though. So the top line is cancer incidence, or cancer cases. The bottom line is our mortality rate associated with cancer. This means we're getting better at treating the disease, okay? That gives us hope. That's what we're looking for. That's why we do what we do. So this is more breast cancer specific related data. And again, I kind of already gave away the takeaway here, but on the left-hand side, you see age, and then you see the risk of being diagnosed, and then the risk of dying from breast cancer. And again, what do you notice? Our risk goes up with age, right? But our lifetime overall risk is about 13%, or 1 in 8 women have the risk of developing breast cancer over their lifetime, right? You're going to know someone who's been affected by this. You yourself may be affected by this. So we want to make sure that we pay attention. So I know it's September now, and it's a reminder that next month is Breast Cancer Awareness Month. And I hope everyone in the room is well aware that we should be getting our mammograms, right? So the current recommendation is screening between ages, starting at age 40 and going through about age 75. Depending on the source and who you ask, every year or every other year. And if you have a family history, or if you have past history with the disease, you may start doing that early. I know I started screening at age 32 because of family history, and I go do it annually. So why is it important to get your mammograms, ladies? Catch it early, okay? What we have seen, so mammograms really came on the scene in the late 80s. A lot of big adoption and just went into the 90s. And what we saw was a dramatic increase in diagnosed cases, okay? Not because more women had breast cancer, but because we were diagnosing women who would never have been diagnosed otherwise, right? But the bigger part of this is that early piece. Mammograms, because of early detection, have decreased breast cancer mortality by 1/3 since the 1990s. And our technology is only getting better, okay? So everybody remembers the old school way of doing it. Some of us still go that route, right? But we've also got ultrasound, we've got high resolution imaging, less invasive and better technology. So again, we're getting better and better at diagnosing these cases early. And why do I keep harping on the early piece? Well, because we all also probably know that breast tumors start in the breast. That's how we diagnose different cancer types, is the tissue of origin. And if we catch it early, so this is demonstrating a stage 1. Has anyone ever heard of ductal carcinoma in situ, or DCIS? So that's what we would call stage 0. That means it's within the mammary duct and it's contained. The reason we call it stage 0 is because the likelihood of it having escaped or metastasized even locally is very, very low. So you can surgically remove that tumor with a lumpectomy, and a lot of times you don't need any additional treatment. As that tumor starts to escape out of that duct, go into the surrounding tissues, maybe start to infiltrate into our lymph nodes, right? And then when we get to stage 4, that's when we're talking about distant metastasis. Okay, and what we mean by distant metastasis is it has spread to other organs in the body. Breast really likes to metastasize to brain, bone, liver, and lungs. Okay, so raise your hands if you think that your breasts are vital organs.
Oh, yes.
Yeah. So they're vital to me personally just because we all have vanity, but can we live without them?
Yes.
Absolutely, right? So if we catch it here, and even if we decide to go beyond a lumpectomy and maybe we get a mastectomy, we remove that breast tissue, right? Great, we're okay. That leads to a lot of our survival, which I'm going to show you graphically in just a second. Brain, vital organ, liver, lungs, yep. Once we get to that point, it makes it much more difficult to treat because, again, you're impeding actual vital organ function. And once the disease has spread, it's much more difficult to go find individual cells throughout the body and make sure that we've killed them than it is to remove a solid tumor, okay? So again, I will say it over and over again, but that early detection is super critical. And again, this is 5-year survival rate for women after diagnosis. So 5 years after initial diagnosis, if you were diagnosed with stage 0 cancer, ductal carcinoma in situ, 100%, right? Again, we can make sure that we get all of that disease out. Stage 1, stage 2, there are substages in here as well, and it's not as clean cut as this appears. But again, as you get down to the more distant metastasis phases, your rate of survival decreases. Okay, again, because that recurrence increases, resistance increases, and the ability to treat those tumors is very difficult. Right? So let's talk a little bit about what cancer even is before I get into a little bit more about my research. Cancer is not new, right? It's not something that we've liked to talk about for a long time. I grew up in rural North Carolina, and I can tell you my family did not like to say the C word out loud, right? It was something that we did not talk about. But it's not new. The first documented cases that we know about were written down by the ancient Egyptians in 3000 BC in this papyrus right here. And not only did they talk about it, that they understood what cancer was, that it was a disease, that they understood that it spread, that they understood and documented how they treated it by cutting it out or by burning it out. But they also understood when it had gone past the point of being able to kind of help someone survive. And these are mummified remains that have been found with indications of bony tumors. So again, we know that this is not anything new. And if you wonder where the name comes from, we can thank the Greeks for that. So carcinos or carcinoma is where we get the word carcinoma. It's Greek for crab, and that's Hippocrates who came up with that because he thought that the body of a tumor looked like the body of a crab with the legs kind of extending out into the surrounding tissue. And oikos means is where we get the derivation for oncology. And I have this quote because again, we're talking about 300 BC, and folks knew things, right? Hippocrates even said, what medicine does not heal, the knife often cures. What the knife can't cure, surgical removal. Cautery often heals. So we could relate that to like radiation treatment, right? And if all of these fail, the disease is incurable. So again, you can learn a lot by looking back at history as well. But if I asked you, what is cancer? Right? I'd probably get a bunch of different definitions. But it's really a group of diseases. So when we say cancer, they're not all the same. There's a lot of variation and variability. But the thing that they have in common is uncontrolled cell growth. Okay? And the ability for those cells to metastasize or move away from the primary tumor. And again, that's typically where we see most of our poor outcomes. We classify tumors by the tissue of origin, so you've got lung cancers and pancreatic cancers and breast cancers. They all share some same characteristics, but what drives them is very different. We'll talk about that with treatment in just a minute. And then you've heard these two words as well, benign. Yep. So benign, they're mostly normal looking cells. They're just growing too much, right? We tend to think of benign as being less harmful. That is typically the case. However, if you have a benign brain tumor, do we need to pay attention to that?
Yes.
Yeah, because our brain's enclosed in that skull, right? And even if a benign tumor is growing, it's going to put pressure on organs and can cause some harm there. Malignant is whenever, again, those become more abnormal and gain that ability to metastasize or move throughout the body. Not all benign tumors will become malignant, okay? But they can. So a lot of times we like to follow them, and sometimes your doctor will tell you we need to just keep an eye on this, we don't need to remove it right now, but then sometimes we do. So great, we've kind of given a definition, but what causes cancer? So I mentioned mutations before, and that's really what this gets down to. So here's the problem with cancer as related to maybe some other diseases. If you have an infection and you go to the doctor, you've got some kind of microbial infection, you go to the doctor, what does he give you?
Antibiotics.
An antibiotic, right? They work great. Why? because it is a foreign organism that is in your body. Okay, it is not human. Cancer are our cells that have gained mutations and now they're growing out of control. Okay? This makes it much harder for us to treat because if they're using normal cell functions, they're just using them differently. So that's just something to keep in mind. Is it almost feels like a betrayal, right? Like my body has betrayed me because these are my cells and they're just out of whack. Okay, they're out of control. So mutations just mean a substitution in your genetic information in your DNA that cause those genes to malfunction. They're not working properly. And there's a bunch of different kinds of mutations. We're not going to get into those. But some of these I did want to just mention is that you can have inherited mutations. So that means that you've gotten a bad copy of a gene from mom, dad, or both. These tend to be a higher risk factor for developing more aggressive forms of cancer because you gain those mutations at conception. When you are a single cell, you have that mutation. So every time that cell divides, it passes that mutation to your other cells. So every cell in your body now has that mutation. So you're at a much higher risk then for gaining additional mutations that may ultimately lead to an uncontrolled cell growth. If you don't have that, you can get mutations just by your DNA needs to make copies of itself to make new cells. Sometimes it makes mistakes. A lot of times those mistakes are fixed, no problem. As we age, our ability to fix those damages becomes a little bit less and a little bit less. So again, another relationship of how these mutations tend to gather over time with age. And then environmental is probably the one we think about the most, right? You guys live in Southwest Florida. What do we have a lot of down here? Sunshine. I'm a redhead. I'm not meant to live in Southwest Florida, right? I have to hide from the sun. But those UV rays, they cause DNA breaks. So my body has to repair those all the time. And the more damage I cause, the more likely I am to get a damage that doesn't get repaired properly. So UV radiation, chemicals in our environment, in our diet, and even infectious agents. So have you heard that human papillomavirus or HPV is associated with cervical cancers and some other reproductive cancers? That virus inserts itself into our DNA, and if it inserts itself in the wrong place, it can interrupt gene function and lead to uncontrolled cell growth. But not all mutations lead to cancer. Not all mutations lead to increased risk of cancer. Mutation in and of itself is not a bad word. Okay, we need mutations in order for us to be diverse, to protect us from other things in the environment, right? So we need them. It's just when we get a few too many and in the wrong spot, it can cause problems. So I teach an entire 15-week course on cancer, and we focus on these 6 hallmarks of cancer for the whole time. We're not going to do that today. But what I did want to point out here is that we as cancer researchers have learned to understand while cancers are different and they're a collection of diseases that have the characteristic of uncontrolled cell growth, there's a lot of variation, but they seem to have a few things in common. They all have too much growth signal, and they're not reliant on outside things to tell it when to start growing. And they are able to evade the stop signal, right? So the way I talk to my students, I say it's like your gas pedal in your car is stuck down and somebody's cut your brakes, right? You can't stop. Now normally those cells would then undergo cell death. So that cell would say, something's wrong with me, I'm gonna go just die so that we don't pass this on, right? Cancer cells have figured out a way to turn that off.
They
Override it, right? The fail-safe is gone, okay? And in addition to that, normally our cells, so who in here loves the fact that we age? Not a fun topic, right? But the reason we age is it's a protective mechanism. So every time our cells divide, the ends of our chromosomes get a little bit shorter. okay? And we have something that is kind of like an internal clock that says you've replicated enough times, you need to stop replicating. Why is that a protective measure? Well, because the longer those cells are around and the more time they replicate, the more likely they are to have gained mutations and errors in their code, and every time they replicate, they're passing that on to the other cells. So if we put a finite number on that and say you can only replicate this many times, it decreases the likelihood that we're going to gain enough mutations to lead to cancer. Cancer cells turn that switch back on and extend the chromosome ends, and now not only do they evade death and they proliferate too much, but they can live forever. They can keep replicating indefinitely. I have cell lines in my lab that we've been growing continuously, and I'm not saying we, but the community, right? Since the 50s, 60s, right? Anybody ever heard of Henrietta Lacks? HeLa cells. So I have some of her cells in my lab. She passed away in the late 40s, early 50s, and again, those cells are growing all around the world. There are more of her cells alive today than were alive when she was alive, right? Because they can grow forever. And the other two pieces of here, we've talked about the ability to invade and metastasize, so they turn that ability on. But then they also have this ability to create and recruit new blood vessels. And this is really important for solid tumors because in a normal body, your tissue can only grow so big, it's pretty small actually, without having to have new blood vessels to bring nutrients to those cells and to get rid of waste. These cancer cells send out signals that say we need the blood vessels to come. So the blood vessels bring all those beautiful nutrients that they need to grow and divide, and provides a way for them to escape and go throughout the body, right? These are all normal functions. These are all, your cells have to be able to do this, your normal cells. So again, it makes it harder for us to treat, but the more that we know about how these cells are working and what they're using to promote proliferation and metastasis, each one of these provides us a mechanism to target for therapies. So we have drugs that stop this signal so it can't form new blood vessels. We have drugs that stop the telomeres from extending so that it limits their production, right? So we can hit each one of these and make better therapeutics. Okay. I'm gonna skip a little bit. I'm happy to kind of come back to this, but in terms of therapies, we've come a long way, but at the same time, we still rely on some of the fundamental things that folks 3000 BC were still using, right? When in doubt, cut it out, right? Surgery is really our primary defense when we're talking about solid tumors, and again, when it's in a tissue that we don't need in terms of a vital organ, it's an easy place to get to, this is a great route to go. But it only works if you're talking about solid tumors and not hematological or blood cancers. We can't really cut out that type of thing. Radiation, discovered this right in the late 1800s, started using it pretty extensively in the early 1900s. This is, you are in effect taking radiation and directing it to a very specific point in the body, causing more damage than the cells can overcome and trying to kill those tumor cells. A lot of times these things will be used in combination, right? In the mid-1900s, we started using pharmacological drugs. So you guys think about that as chemotherapy. All that means is it's a drug that you can either take orally or intravenously. It is systematic or systemic, going throughout all of your body. Why is that a good thing? Well, it can get to those cells. Let's say they've moved around or they've escaped. It should be able to get to them and hit them, right? What's the bad thing? It hits everything, okay? And again, because these cells are our own cells and they're using normal processes, it's very hard to target only the cancer cells. So what most chemotherapies do is it hits anything that's actively proliferating. So it's going to hit the cancer cells more because they're proliferating more. But what happens? Your hair falls out. Why? Because those follicles are proliferating a lot. Your immune system gets compromised because those bone marrow cells are proliferating a lot. right? And so we have to balance how much do we give a person to kill the cancer while also making sure that we don't harm them too much. Here's where we're moving to. The last few decades is targeted therapies, okay? So you may have heard of, if you're HER2 positive or HER2 amplified, again, that's a normal gene, but in that subtype of cancer, they express that protein at really high levels. So we have developed drugs that bind to that protein on those cancer cells, and it's going to shut that cancer cell down really quickly while not really harming normal amounts of protein in other cells, okay? The problem is you have to know a target. And not all cells have that target, but for those that do, much better response, much less side effects. And what we're learning, I'm just gonna say this very quick, resistance, right? If you don't kill all the cancer cells the first time, the ones that survive tend to be resistant. So we don't use a lot of single agents anymore. What we use instead is what we call a chemo cocktail. Not as fun as the cocktails that you have on your table. What this means is a combination of drugs. And it's not just any drugs, it's drugs that hit different mechanisms or different pathways within the cancer cells to try and kill off as many as possible to have a much greater effect, and that you don't allow the cells to become resistant. Okay. The problem is that we need to constantly be building a larger arsenal of drugs. Because cells will become resistant over time, the more drugs that we have with better mechanisms of action, with better targets, the better we're going to be able to treat folks. And we are really getting to a point now with technology where we can personalize patient care. And I don't mean like, okay, all breast cancer patients receive this drug and all lung cancer patients receive this drug. We've been doing that for a while. But even within the same cancer, so let's just say breast cancer, within a specific subtype, so let's say HER2 amplified, even within the same patient, if I have a primary tumor, it's going to have different mutations than a metastasis somewhere else in my body. Okay? We can take biopsies, and we can do molecular profiling, and we can pinpoint what mutations do you have, what pathways are going wrong, what are driving your tumors. And instead of treating everybody the same with the same cocktail of drugs, if you have the green profile, we're going to give you the green drugs. If you have the pink profile, we're going to give you the pink drugs. So that we're not going to have as many off-target effects. We're going to have a greater efficacy of that drug, and hopefully we're going to help those patients not only have a longer life, but a better quality of life as we move forward. All right. So here's a little bit about what I do. So I came from Tulane, School of Medicine there. I worked within a cancer center, right? So I was surrounded by other cancer people all the time. And then I moved to Southwest Florida, FGCU, go Eagles, love it. I am the only cancer biologist at FGCU, okay? So it's an interesting position to be in. I still collaborate with a lot of my colleagues at Tulane and other places around the country. But what it's allowed me to do is really get involved in projects that I never would have thought I would have gotten involved in. So I'm really interested, one, in breast cancer, but I'm interested in this subgroup right here. So we call this triple negative. So these folks are negative for the estrogen receptor, negative for the progesterone receptor, and non-amplified for HER2 new. That means we don't have any targeted therapies for this population of patients. And while it only represents about 10 to 15% of cancer diagnoses, it affects younger women, it affects minority women disproportionately, and it is a very aggressive and highly metastatic form of breast cancer, right? And then I'm also very interested in using natural compounds as lead compounds for pharmacological development, drug development. I know sometimes people are like, oh, that's kind of weird. Like, why are you doing this kind of touchy-feely Mother Nature kind of stuff? That's not what I do. Has anybody ever heard of Taxol or paclitaxel? Okay. Yes. So in the 1960s, this is not anything new that I'm telling you. we're looking for anything that's gonna help with cancer and be a cancer treatment. Does anybody know where Taxol comes from? Where it was originally discovered? Trees. Trees. The Pacific yew tree, the bark. So in the 60s, there's this big call by the National Cancer Institute, find a cancer treatment. We don't care where you find it. Test everything. And they did. And from the bark of the Pacific yew tree, they pulled out this derivative, Taxol, and it is still used today as one of the most effective general chemotherapies. Now, we don't go isolate it from the Pacific yew anymore. We can synthesize it in the lab, and that's what I'm interested in doing, right? Finding things in nature, identifying what the active compound is, and then using that to be able to scale up for a better treatment. We've done things with essential oils. I'm going to talk to you a little bit about still beans and then some Eastern diamondback brown snake work. I know, right? So, who likes red wine?
Oh, yes.
Great, me too. Anybody ever heard it's good for you? We at least like to tell ourselves that, right? So it has been, and I started this work quite a few years ago, but there are some natural defense compounds found in plants, including red grapes, which are where we get red wine from, and these are called still beans. If you've ever heard the one resveratrol,
Yes.
that's the one that I started working with, and then we've started doing other things from there as well. There have been a variety of different health benefits proposed with still beans and estrogen positive cell lines. And the hypothesis was that if it's a similar shape and size, maybe it binds to the estrogen receptor and can stop it. Because it's not estrogen, maybe it can inhibit the signaling. So we tested in our cell lines, and it worked. It was pretty interesting. We have a whole library from the USDA of these very similar compounds that just have different groups on the ends here that make it more or less effective. And we were like, okay, we want to make sure, because we think it's binding to the estrogen receptor, let's test it in our triple negatives, because they don't have the estrogen receptor. It was supposed to be a negative control. It was not supposed to work. Turns out it worked better in our triple negative cells than it did in our ER positive cells. So it sent us on this whole kind of rabbit hole. And we've published a couple papers. One where we were looking at the ER positive, and we did show some of our ER negative. So it inhibits breast cancer growth. It induces cell death in those cells. This is one of my past students who did most of that work. We also tested, so if these compounds aren't specific to the cancer cells, if they're just toxic in general, then they're not going to be a very good compound to use. So we have a zebrafish model. You guys know those little stripy zebrafish you can buy at the pet store? We use them in the lab. And we were just looking to see, does it have any effects on normal development of cells? And it turns out at low doses, it did not have any negative effects on those fish, which that's a good sign. And then we also saw that these still beans not only killed the cells, but the ones that didn't die, they started to change their shape. So why is this important? So normally triple negative cells are long and spindly, okay, and that means that they can invade and metastasize very easily because of those protrusions. When we would treat with some of these analogs or these derivatives, the cells started not growing in singles, but growing, touching each other. It started to make more rounded shapes that look more like normal breast cells. So even if they're just reverting back to a less aggressive type, that's very interesting for this subtype of cancer. And then my graduate student tested these in a synthetic migration and invasion assay in the lab, where we seed the cells on top of a membrane, and the cells like to go down to get to food at the bottom. And if we treat with these compounds, it stops them from being able to move through to the other side, right? So that we were thinking maybe it can help us with metastasis, which again, for triple negative is really the goal. She is currently working on going from a 2D model system, so growing cells flat in a dish. Turns out cells don't actually grow on plastic in 2D when they are in your body. So we wanted to have something that mimicked more of what's actually going on. So she's been able to form these 3D tumor spheroids in culture. And she's in the process of testing to see if our effects that we saw on the 2D model were in 3D. So we're getting some pretty interesting stuff there. Yes.
I don't want to cut you off, but
Yep.
can we have people start to
Absolutely, jump up, and I'm almost done.
To minimize disruption, we'll start here and go around this way. So please go ahead.
So I mentioned these zebrafish, and the really fun thing that we're doing now, which has been really a struggle for my student. We've been working on this for about 2 years, and she's ready to pull her hair out, but we think we've got it working. Injecting human cancer cells into the zebrafish yolk sac to use as a metastatic model, okay? So we inject them here, and we're looking for the cells moving, and then if we treat them with our drugs, does it stop it, right? Because again, we want to make sure that what we're seeing in a very artificial system is holding true in a whole animal model before we can go to anything more clinical. So here are the first results. These are kind of hot off the presses. This is her initial injection, and you can see the tumor starting to grow and starting to spread. And this is just only after a couple of days. So she's doing this for longer term to see how many days out she can go, and then we'll start treating with the drugs to see if we can inhibit metastasis that way. So we're pretty excited. And then the last thing I wanted to talk to you about is this really wacky study that we started. Again, I'm a cancer biologist, but I'm always looking for things that might have potential. I work with a lot of herpetology people. They like studying snakes. They like studying reptiles, right? I don't know if you know, but we have a very large eastern diamondback population in Southwest Florida and on FGCU's campus. So they go out, they're tracking them, they're looking at them, they're documenting them. And there's some information in the literature. Snake venom's been used for a very long time in various cultures for different types of diseases. We've used them for anticoagulants and treating blood disorders. There's some information saying that they may have proteins that would inhibit some of the signaling pathways that we see in cancer, right? So well, next time you're out, why don't you guys just start collecting some venom? No. I don't collect them. I get it in this nice little tube, and it's just a powder, and it's just like any other drug that I would ever work with, right? So we have a collaborator, we have collaborators at FGCU, but we also have collaborators up at USF in Tampa. They collect the venom for me, and what they wanted to look at initially was they had collected venom from eastern diamondbacks at different geographical locations across Florida, and they wanted to see if the venoms were any different in terms of their composition, and I tested them to see if there was any difference in impact on our cancer cells. I've done this in a couple different cell lines, and the results kind of hold true, but you can see as we increase our doses, it really quickly drops the viability of these cells. These are 100%, and it drops them down below about 50. They all kind of look the same, but it's all eastern diamondback venom, so that kind of makes sense. Except that when we compared this population here in the panhandle, we had babies, so juvenile snakes, and we had the adult snakes. And it seems like the adult venom is more potent and able to kill them at a lower dose than the juveniles. So actually next week I'm going up to USF, I'm taking samples, those exact samples, and sending them up to colleagues at FSU to run component analysis on them. So they'll actually separate the venom by individual protein. They'll send those back to me, and instead of looking at the crude venom, we'll test each individual protein to see which one is having the effects that we're seeing. That's where we get to the lead compounds, right? If we can identify specific proteins. So why are my reptile friends interested in going out and collecting venom for me to look at? Why does that help them? Nobody here cares if we save the rattlesnakes. Yeah. Not too many of you. But if the rattlesnakes could cure cancer, would you care about saving the rattlesnakes?
Yeah.
Right? So I get to do these really fun collaborative projects with colleagues across the state to look at, what can we do, and it's really beneficial to both. So with that, my real takeaways for you is that cancer is complicated. We've been doing this for a long time. As technology has gotten better, we've learned a lot through our research, and we are making significant progress, right? Early detection, early detection, early detection. But here's the real thing. Even if we don't have a cure, and that's really not the goal, we're never going to find a magic bullet, one single cure, because cancers are so diverse, right? Even person to person. we are getting better therapies the more that we learn. We are extending lifespan, and that gives patients longer time for us to develop better therapies, right? So again, if we can extend quality of life and timeline, we have a chance to continue to do better work. And then just the last thing I'll let you guys know, if anybody's interested in this, myself and 3 of my colleagues at HECU have gotten a grant through the National Science Foundation to look at female representation in STEM and leadership, right? So not just biology, but across the board. And we are having our second day of dialogue coming up at the end of this month to look at data within HECU and how we compare nationally, and do we have places where we could do some extra work in terms of gender, but also other intersectional areas like race and ethnicity. So if anybody's interested in that, you can learn more. I'll give you the website, or we can chat later. But I thought that might be of interest to this group. But with that, I'm happy to answer any questions. My only disclaimer is I am not a clinician. My grandma used to call me and tell me, such and such has cancer, what should they do? And I said they should go to their oncologist.
Do doctors or oncologists, cancer doctors, do they always look for new treatments? Are they in the circle of new information?
So here's the kind of balance and plus and minus that we have. We want to make sure that we're bringing new therapies to the market, right, to the patients. But we also want to make sure that we're not doing harm. We're not causing more harm than good. So the timeline from a new therapy being developed in a lab to getting to the clinic where it's actually being used broadly is pretty long. It can be 10 years.
Yeah.
And there's been some negatives to that. There's been a lot of kind of negative press around that, but at the same time, we also know when we roll out things too early and then we don't see the efficacy, then the trust in the scientific community gets eroded, and we don't want to have that happen either. There are a lot of clinical trials that are ongoing, but I know every time I used to go to see my my gynecologist would be like, hey, what's new? What's new with breast cancer, right? And I would kind of keep them in the loop, but it is definitely a partnership between kind of academia and the clinical world, because they're very distinct roles, and unfortunately our physicians don't have time to really dive into the literature every day and find out what's new, because it's a lot. There's a lot going on.
I have one more question. Bee venom, have you used bee venom to test it?
I haven't. I would love to. The problem with bee venom is you need a lot of bees, because they only have a little bit of venom. But I know that there has been some research done on that. Snakes are pretty easy, especially these big ones. They'll give you, they're happy to give you a lot of it. Yeah. Yes, ma'am.
Quick question, we know that, or I've heard that the vitamin D3 with the K2 is good for women to take, but would we also want to then add maybe resveratrol as a supplement?
So here's the thing, and I know I just gave you this whole big spiel about how great resveratrol is.
Right, like what quantities?
So the thing that I was showing you was not actually resveratrol. The ones that we found to be most active were derivatives of resveratrol, so there's been little tweaks to it. The problem with resveratrol is it's not very bioavailable, so once you digest it and it goes through the first pass with the liver, it's basically gone, right? So part of this would be developing a more stable molecule that is, and then you have to talk about delivery. Is it oral? Is it an injection? Is there kind of how do we get it to the cells? I haven't seen anything that says it's harmful to take resveratrol supplements. But I had a slide that showed like how much resveratrol was in different kinds of wines. But the problem there again is it's such small amounts that you have to balance alcohol consumption with your resveratrol amounts. And the thing is, like, drinking is a risk factor.
Yes.
So 1 glass a day is not going to hurt anybody. But when you get to 2 or 3 a day, now you start to see your risk factors go up. Smoking, obesity, just inactivity, all those things can lead to increased risk. Yes, ma'am.
Oh, sorry.
Yeah.
So I started my career as a functional medicine practitioner, and I worked with some clients with the Cancer Society back in home. One of the areas that I thought, and I missed the first part of your presentation, was about gut health, about the microbiome, and what we found with anyone at all, because you can take a lot of things, but it's the body's ability to absorb.
Right.
And so that is so fundamental. You know, when we're
Yep,
talking about how much and so that was one. And also with people in terms of post chemo or something like that, it just completely, we're talking about microbiome and our gut health. And so in terms of your research, in terms of anything you're doing, is that in play at all? Or?
So, I mean, there is a lot of research out in terms of gut health and not just breast cancer, but many different types of cancer and absorbance of drugs and things like that. One of the problems with academia is, we have limited bandwidth, and so like each of us become very niche, and then it's a practical thing of putting it all together, which is more difficult. But I do know that there's been a lot, and we're even investigating, and I know we're eating dinner, but like fecal transplants, especially post chemo, when you're basically wiping everything out to reseed so that we make sure that we have good health. There are supplements that you can take that will help support, and then diet in general, right? Like food is medicine, period. We don't like to think about it that way, but when I'm doing natural compound work, it's the same thing. We can learn a lot from other cultures. We can learn a lot from just paying attention, right? And so I think that is a key part. But everybody's different. So even the genes that you have in these different places are going to interact differently with your microbiome. So there's no one size fits all, which makes it a lot more difficult.
But it's a lot about your body's ability to absorb.
Yep.
And I think often that we talk about medicine or we talk about things, and that piece I think often is missing.
Yeah, absolutely.
I think we can take one more question.
I'm happy to talk to anybody who does have another question later.
I was curious because you had said environmental is a big part of, potential cause. Is that part of your research as well? Have you been able to, whether it's fertilizer or them spraying us with mosquito toxins or food, if we eat non-organic food, have you seen anything with a higher risk that we should be avoiding?
So it's very hard because the problem is that none of us live in a bubble and are only exposed to one or two things, right? So it's very hard to draw that line. Now, epidemiological studies, that is what they do in the public health sector, is look for these hot spots, right? And I mean, there have been correlations with, Roundup causes cancer. Yes, if you're around it all the time, right? If you're working in agriculture and you're spraying it all the time, absolutely, there has been a link there. If you use it once a season around your house and you're not necessarily getting a lot of light, it's probably okay. Same thing, plasticizers. We drink out of plastic gets warm. Okay, well, we know that we've taken BPA out of everything. I don't want to be a Debbie Downer, but if you take BPA out, guess what you have to put in? Another plasticizer that we don't know what it does, right? So it's not that, we've solved the plastics problem. We just don't have enough data yet to know what the issue is. I don't want anybody to leave here being like, it's inevitable that we're all going to die and there's nothing I can do. That's not what I'm saying. I know. It's quality of life and paying attention. You still have to live your life, right? I eat red meat. I don't eat it every day. Right? I know. I drink wine. I don't do it every day, right? And so you just have to kind of balance it out because you want to make sure that you are paying attention and changing what you can, but that you're not just being miserable and being so hyper aware that you're not really living your life. So that would be my advice. And then go to the doctor. Do not ignore things. If you think something might be wrong, ignoring it's not going to help. Right, so go and get checked out. When I worked in New Orleans, we had, and even back home in North Carolina, there was a cultural stigma around talking about it and acknowledging it. It was kind of like, if we just ignore it, it'll go away. No, if you just ignore it, it's going to get worse if it is something. And if it's nothing, guess what? You go and you have that little bit of stress, and then you have that relief, right? That everything's okay. So that is my PSA for today.
And Christine, you had a question.